So this is about this one. So coming to the module 6, that is fatty acids and lipids. So in this module we'll be studying about the classification of fatty acids as well as classification of lipids. In addition to this, we'll be studying only the formation of mevalonate from acetyl CoA. This is comes under metabolism. So in fatty acids, first we'll be discussing about the classification of fatty acids, their structure, properties, function, and metabolism of fatty acids. Then classification, structure, properties, and biological functions of simple lipids, triacyl glycerols, and waxes, and compound lipids such as phospholipids and glycolipids, cholesterol, which is a derived lipid, structure, properties, and importance. And the last one is the eicosanoids. Eicosanoids are the carbon 20 containing uh, the long chain of fatty acid derived hormones. These are also known as the local hormones. And this is the syllabus given in the module 6. And we know that to understand the membrane transport, first of all, to understand the membrane transport, how the molecules move across the membrane, because biological membranes are lipid bilayers, to know how the molecules go out or they come in. To understand this, we need to understand the structure of the biological membranes, and to understand the structure of the biological membranes, definitely one should have the thorough knowledge of the lipids. So in Greek, lipos means fat. So these are the third major group of biological molecules found in the cell. And lipids are not polymeric like the polysaccharides and the proteins, because in the case of proteins they are built with many amino acid residues, whereas in the case of polysaccharides they are also built with many monomeric sugars. But whereas lipids are not polymeric in nature, but they can form various aggregates like biological membranes, micelles, the liposomes, and so on. And these lipids are hydrophobic or amphipathic in nature, and they have many functions we'll be discussing in the next slide. But three important functions are these lipids are involved in the formation of biological membranes. In the sense, biological membranes are formed from the lipids, and these lipids are also serve as energy stores, and some lipid derived molecules are involved in the signaling events, mainly the phosphatidylinositol triphosphate and phosphatidylinositol diphosphate, and so on. And coming to their presence, their occurrence, these lipids are present in humans, animals, and plants and microorganisms. And we know some lipids are animal origin, some lipids are plant origin. The animal fat, egg yolk, butter, and cheese, all these are lipids of animal origin, whereas the vegetable or cooking oil and vanaspati, all these are the lipids of plant origin. And coming to its medical and biological importance, the I think, as I already said, that the lipids have many functions in the human body or in living organisms. And the first and important function is they are the source of energy. So the lipids are the source of energy, in the sense as we know that each gram of fat can release 9 kilocalories of energy. And lipids are also mainly the triacyl glycerols are the ideal form for storing energy in the human body. And different class of lipids, mainly phospholipids and the sphingolipids and the steroids, all these are the components of the cell membranes or the biological membranes. And some lipids are hormones. For for example, steroid hormones and prostaglandins. Most all these steroid hormones are derived from the cholesterol, and both male and female reproductive hormones are the steroid hormones, and they are derived from the cholesterol, that is the derived lipid. And the lipids are also essential for the absorption of fat soluble vitamins, and the lipid present under the certain organs can act as serve as the shock absorber. As a result, they protect the vital organs, and the lipid that is present under the skin can also act as a thermal insulator. That's why the certain cold animals living in the extreme cold condition, like polar bear, can survive under extreme cold condition because of the presence of the this fat under the skin. So because that acts as a thermal insulator, and and of course the fat that is present around the kidney serve as a padding which prevents the against the injury. And then coming to as I already said that the lipids are also components of the cell membrane and also the nervous tissue. And some lipids are some simple lipids are the precursors for the synthesis of complex molecules like cholesterol, and the lipoproteins. The lipoproteins, they are the storage, or they are the transport vehicles for transporting lipids from the liver to the extrahepatic tissues, or the extrahepatic tissues to the liver. So that means the lipoproteins are also, they are composed of lipid as well as protein. They are involved in the transport of lipid in the blood. I think many of you have already known that VLDL, IDL, LDL, HDL, all these are lipoproteins. They are involved in the transport of lipid in the watery blood um, from the liver to the extrahepatic tissues, or the extrahepatic tissues to the liver. So mainly the HDL is a good cholesterol. It is mainly involved in the transport of the transport of the lipids from extrahepatic tissues to the liver for the degradation. And also LDL transport the lipid from the liver to the extrahepatic tissues. And and in addition to this, some lipids are, as I already said, that they serve as hormones. And as I already said, that the eicosanoids, carbon 20 containing long chain fatty acids, these eicosanoids, they are mainly the they are mainly these prostaglandins, prostacyclins, thromboxanes, and leukotrienes. All these are the eicosanoids. They are derived from the fatty acid, unsaturated fatty acid, arachidonic acid. So these are all these eicosanoids have various physiological physiological functions in the human body. And as I already said that and the lipids that are present on the myelin sheath also acts as an insulator that propagates in nerve signal propagation. And we can also see here, this is the neuron, and you can see here, this is the myelin sheath, and this myelin sheath prevents the leakage of the action potential during action potential propagation. And this myelin sheath is rich in the lipids. And some saturated fatty acids have antimicrobial and antifungal mechanism, and that means they are some saturated fatty acids are antimicrobial antifungal agents, and some saturated free fatty acids are pheromones, like they are the sex attracting substances in animals like a tiger. And finally, as I said that some lipids are also lipids or lipid derivatives, they are also involved in the signal transduction pathways, mainly the phosphatidylinositol diphosphate and phosphatidylinositol triphosphate, and so on. So these are the major functions of the lipids in the body. And as I already said that, and you can also see here, this is the this is the myelin sheath, and in case of this myelin sheath has 70% of lipid, only 30% of the protein, and this in case of multiple sclerosis, and in multiple sclerosis it is a disease, and in that multiple sclerosis the myelin sheath gradually deteriorates. As a result, the bare axons cannot transmit the action, conduct the action potential or the nerve impulses. And so that means the myelin sheath that is present on the nerves prevents the leakage of the action potential. And as I already said that all these the male and female reproductive hormones, all these are derived from the cholesterol, which is a derived lipid. You can see here all these the progesterone, corticosterone, and aldosterone. All these are testosterone. All these are derived from the cholesterol, which is a derived lipid. So that means both male and female reproductive hormones are synthesized from the cholesterol. And then uh, then coming to, as I already said, that all these are the eicosanoids, they are prostaglandins, prostacyclins, thromboxanes. All these are derived from the arachidonic acid, which is a carbon 20 containing long chain fatty acid. They have various functions in the body. And then coming to the chemical nature of lipids. The chemical nature of lipids, we know that the lipids are the soluble only in the organic solvents like chloroform, but they are insoluble in the water. And these lipids are classified in various ways, but Bloor has proposed the classification of lipids into three categories. One is simple lipids, second one is compound lipids, third one is derived lipids. And in simple lipids, we have fats and waxes. In the compound lipids, we have the glycerophospholipids and the sphingolipids. And in the derived lipids, mainly the steroids. So, since all the lipids are the esters of fatty acids and glycerol and other nitrogen containing compounds. So first, in order to understand the classification of lipids, first we need to understand the fatty acids. So we know that fatty acids, they are the long chain monocarboxylic acids. They have, you can see here, and they have the one side hydrophilic carboxyl group, and other side hydrophobic hydrocarbon chain. So that means they have one side hydrophilic carboxyl group and other side hydrophobic hydrocarbon chain. So they are the very long chain fatty acids. And long chain carboxylic acids. And these fatty in the human body, more than 100 fatty acids have been identified in various species, but all these fatty acids differ in their length and also the double bonds. And most of these fatty acids have the pKa around 4.5 to 5. And these free fatty acids are based on the chain length. These fatty acids are classified into short chain, medium chain, and the long chain fatty acids. And the short chain fatty acids are liquids at room temperature, and medium chain and fatty acids have 6 to 10 carbons, whereas the long chain fatty acid, they have the 12 or more carbons. And most of these fatty acids are also present in foods. And based on this is based on the tail, the length of the carbon length, whereas the based on the degree of saturation. Based on the degree of saturation, these fatty acids are also classified into two types. One is saturated fatty acids, second one is unsaturated fatty acids. So in saturated fatty acids, you can see here they do not have the any double bonds. They have only the carbon-carbon single bond in their structure, whereas so that's why, since they have the higher melting points and they are more stable. Whereas in case of unsaturated fatty acids, they have the one or more double bonds in their structure, and if they have only one double bond, they are known as the monounsaturated. If they have more than two or more double bonds, they are polyunsaturated fatty acids, as the unsaturated fatty acids are liquids at room temperature, and they have the lower melting point, and they are less stable. And you can see here that based on the shape, and these in case of unsaturated fatty acids, depending upon the based on the nature of the double bond, in the double bond, if if the two hydrogen atoms across the double bond present on the same side, then it is known as the cis fatty acids. If the two hydrogens across the double bond are present on the opposite side, then it is known as the trans fatty acid. And you can see here, when there is a when the cis double bond is present in the fatty acid chain, that creates a bend or the kink in the structure, and whereas compared to the trans. So when the fatty acids have when the fatty acids have the straight tails, and they pack very close very tightly, whereas when the fatty acids have the kinked tails because of the cis double bond, and they are fluids at room temperature. And now coming to the fatty acid, how do we number the fatty acids? So in generally we can number the fatty acids. The carboxyl group gets 1 from the carboxyl group. We number the fatty acid from till the last the methyl group, whereas the another way of numbering is the that is the carbon that is far away from the carboxyl group, that is also known as the omega carbon, because in Greek omega means that is the last letter, that's why it is known as omega carbon. And so usually when we represent the fatty acid with structure or the position of the double bonds, we number from the carboxyl group, we should give the number 1, and so on. And so these are some of the list of unsaturated and unsaturated fatty acids. You can see here the carbon 12 containing 14, 16, 18, 20, 22, 24, and all these you can see here the lauric acid or myristic acid, formic acid, stearic acid, arachidic acid, behenic acid, and lignoceric acid. All these are saturated fatty acids, and in all you can see here that with increasing the number of carbons, you can clearly notice that the melting point also increases as the tail length increases. So that is the number of carbons increases, the melting point of the fatty acid also increases. That is in case of saturated fatty acids. Whereas in case of saturated fatty acids, whereas in case of unsaturated fatty acids, you can see here that the carbon 16 containing unsaturated fatty acid is the palmitoleic acid. So this is palmitoleic acid has one double bond, whereas the oleic acid also has one double bond, but the 18 carbons, and the linoleic acid has two double bonds and the 18 carbons. Linolenic acid has the three double bonds and the 18 carbons, and the arachidonic acid has four double bonds and 20 carbons. You can notice here that as the double bond increases, of course, with increasing the carbon chain length, and in case of saturated fatty acids, the melting point increases. But whereas in case of unsaturated fatty acids, with increasing the number of double bonds, the melting point decreases. And that means unsaturated fatty acids have lower melting point compared to the saturated fatty acids. And this you can saturated fatty acids. And this you can also see here, and you can see here, this is the number of carbon atom, that is the carbon chain length, and this is the melting point. And you can see here that if the, for example, you can take here in this case, if the 14 carbon containing saturated fatty acid and unsaturated fatty acids, so compared to the saturated fatty acid has compared to saturated fatty acid, unsaturated fatty acids have the lower melting point, having the same carbon chain length. But among the unsaturated fatty acids, compared to the trans double bond and the cis double bond has the lower melting point. As I already said that In case of saturated fatty acids, they have the straight tails, and the unsaturated fatty acids, the presence of the double bond creates a kink or the bend in the structure. Mainly the cis double bond always creates a kink in the structure. That's why when we write the phospholipid or the we generally write like this, in the sense it indicates that the unsaturated fatty acids are present in the phospholipid, and that's why these unsaturated phospholipids that are present in the biological membranes are responsible for the fluidity of the membrane. If the membrane has the all saturated fatty acids, they pack very tightly, they very closely. So as a result, the movement of the molecules across the membrane is not possible. That's why only that is why the membrane fluidity also increases with increasing the degree of increasing the unsaturation of the fatty acids. And so from this we can understand that the saturated fatty acids have the higher melting point, whereas the unsaturated fatty acids have the lower melting point and lower melting point. In case of two unsaturated fatty acid having the same carbon atoms or the same carbon length, if the one is a trans and another one has a cis double bond, the fatty acids having the cis double bond has a lower melting point, because cis double bond creates the bend or the kink in the structure. And most of the fatty acids that are present in the biological system contain even number carbon atoms, that is typically from 14 to 24, and but carbon 16 and carbon 18 containing fatty acids are most common. Then, as I already said, that the configuration of the double bond in most of the unsaturated fatty acid is cis and cis, and the so then, as I already said, that with increasing the carbon chain length, the melting point increases. With increasing the number of double bonds, the melting point decreases, and this is the this you need to remember this one. And as I already said, that with increasing the unsaturation, the fluidity of the fatty acids also increases. So that is the that also important here. And then coming to the, and of course you can also see here all these saturated fatty acids, they have the straight tail, and whereas the here unsaturated fatty acids, they have the kinked tail because of the presence of the cis double bond. And you can see here as the number of as the you can see here the saturated fatty acid, they pack very tightly, and whereas unsaturated fatty acids cannot stick together because of the bend or the in the structure in the structure. And of course you can also see here as the number of double bonds increases, the tail becomes more and more kinked. Here oleic acid has only one double bond, and whereas the stearic acid and the palmitic acid has no double bonds, they have the straight tails. And this is in case of oleic acid, one double bond creates a bend in the structure, and whereas as the number of double bonds increases, in this case two double bonds, whereas in this case three double bonds, whereas in this case four double bonds, as the number of double bond increases, the fatty acid becomes more and more kinked. As a result, the fatty acids also become fluids, more and more fluids, with increasing the degree of unsaturation. And this is about this one. As I already said, that these unsaturated fatty acids, when they are present in the biological membranes, they can also increase the fluidity of the membrane. As I already said, that if the saturated fatty acids are present in the membrane and they pack very tightly, as a result the movement of the molecules is not possible. Whereas in case of unsaturated fatty acids, they pack very loosely, and so that's why the unsaturated fatty acids increases the fluidity of the membrane. And in addition to this, the cholesterol, that is a bulky molecule, that is a derived lipid, when it is present in the phospholipid, in the between the phospholipid molecule, that decreases the close packing of the phospholipids. As a result, the fluidity of the membrane also increases. And of course, now coming to the essential fatty acids, as some fatty acids are essential for the growth of the organisms, or the so these fatty acids are mainly the linoleic and the linolenic and the arachidonic acid. And as I already said, that these linoleic and the linolenic arachidonic acid, mainly the arachidonic acid is the precursor for the synthesis of various Eicosanoids, those are local hormones, means the prostaglandins, prostacyclins, thromboxanes, and the leukotrienes. All these are local hormones. They have the various functions in the human body. So, in addition to these fatty acids, there are some unusual fatty acids present in certain organisms. Those are branched chain are the ring containing fatty acids. For example, tuberculous acid is mainly present in the microorganisms, whereas the lactobacillic acid is found in the certain lactobacillus organisms. So these in addition to this, these branched chain fatty acids are also present. These branched chain fatty acids in general increases the fluidity of the membrane. Fluidity of the membrane. So this is about the fatty acids. So now coming to the classification of lipids. As I already said that lipids are Bloor has classified lipids into three types. One is simple lipids, second one is the compound lipids, third one is derived lipids. And first we will discuss the simple lipids. So simple lipids, there we have, they are also, they are the esters of fatty acid with alcohol. You can see here, this is the alcohol, that is a glycerol, and this is the when the glycerol, when the 3-hydroxyl group of the glycerol esterified with the three different fatty acids, you can see here. Then the then the fats are formed. In the sense, fats are nothing but the esters of the glycerol and the fatty acids. And here, if the three fatty acids that are esterified with the glycerol are of the same type, then it is known as the simple triacyl glycerol. And if the three fatty acids are of different type, then it is known as the mixed triacyl glycerol. That means the fats are nothing but the glycerol with fatty acids esters. So they are also known as the triacyl glycerols. So now you can also see here, this is the glycerol, and when the three fatty acids are esterified and three water molecules will be removed, as a result the triacyl glycerols are formed. As I already said that if all the three fatty acids are of same type, then it is simple triacyl glycerol. All the three fatty acids are of different type, then it is a mixed triacyl glycerol. And you can also see here, if it is yeah. Then coming to the waxes, these waxes are again esters of fatty acids with long chain alcohol. So here we have the lanolin or the wool fat, or this second one is the beeswax. And you can see here in this case beeswax is an ester of palmitic acid with methyl alcohol. You can see here, this is the for example of the wax. In here the oleal alcohol is esterified with the stearic acid in this structure. This is a very long chain alcohol, and it is esterified here with the stearic acid. So the waxes are the esters of the fatty acids with very long chain alcohol, whereas triacyl glycerols or the fats are the esters of the glycerol with fatty acids. And this is about the waxes and the fats. Now coming to the compound lipids. So in compound lipids, so these compound lipids are the esters of fatty acids with alcohol containing nitrogenous bases and also the additional groups. And these compound lipids are further classified into the glycerophospholipids or simply phospholipids, and also the sphingolipids. And these glycerophospholipids, in the glycerophospholipids, the alcohol is the glycerol, whereas in the sphingolipids, the alcohol is a sphingosine. As we already said that in all lipids, either the fatty acids are esterified with the one or other alcohol, either glycerol or since other alcohol. Whereas in this case, in case of phospholipids or the glycerophospholipids, the alcohol is glycerol, whereas in case of sphingolipids, the alcohol is the sphingosine. And these compound lipids are also divided into in other way, that is phospholipids or the glycolipids. So first we will discuss the phospholipids or the glycerophospholipids. In the phospholipids or the glycerophospholipids, as I already said that here they are the esters of the glycerol with fatty acids and also the other nitrogen containing substances. substances. Here you can see here this is the glycerol and the 3-hydroxyl group. It has a 3-hydroxyl group and the 2-hydroxyl group. The first and the second 2-hydroxyl groups are estrified with the 2 fatty acids. You can also notice here this is the glycerol and it has a 3-hydroxyl group, one first carbon, second carbon, third carbon. The first hydroxyl group is estrified with one fatty acid, second hydroxyl group is estrified with another fatty acid, and the third hydroxyl group is estrified with the phosphoric acid. So this structure is known as the phosphatidic acid or phosphatidate. Phosphatidic acid or phosphatidate. So when this phosphatidic acid is further estrified with when this phosphatidic acid is further estrified with the other compound, other nitrogen-based compound, then we get different types of phospholipids, and these different types of phospholipids are the phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, and phosphatidylinositol. You can see here that again. So the phosphatidic acid, this is the structure of the phosphatidic acid. So this is the glycerol, as we already said that these are the 2 fatty acids, and the third hydroxyl group of the glycerol is estrified with the phosphoric acid, and this structure is known as the phosphatidic acid. When this phosphatidic acid is further estrified with the serine, then it is known as the phosphatidylserine, and it is estrified with the ethanolamine, then it is known as phosphatidylethanolamine. With choline, phosphatidylcholine, inositol, phosphatidylinositol. With the glycerol, is the phosphatidylglycerol. So this that means the phosphatidic acid, when it is estrified further with the choline, it is known as the phosphatidylcholine. If it is with ethanolamine, it is known as phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, and phosphatidylglycerol, and so on. So this is about the phospholipids, and you can see here in this phospholipids, what you are seeing here is that these are the two fatty acid tails, and it is the head group. So when we represent the represent the phospholipid structure, you should understand that this is the head group, whereas these two are the fatty acid tails that are present in the phospholipids. So this is about the general structure of the phospholipids. Now, as I already said that we will discuss one by one, and the first one is the phosphatidylcholine. As I already said, that when the phosphatidic acid is further estrified with the different nitrogen containing compounds, we get the phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, and phosphatidylglycerol, and so on. So first one is the phosphatidylcholine, and you can see here that here in the phosphatidylcholine, and this is the glycerol, and the first and the second hydroxyl group of the glycerol is estrified with the 2 fatty acids, and the third hydroxyl group is estrified with the phosphatidic acid, which in turn estrified with the choline molecule. As a result, it forms the phosphatidylcholine, and this phosphatidylcholine is also known as the lecithin, and it is the major phospholipid present in the biological membranes, and it is also present in the egg yolk and lipoproteins. So this phosphatidylcholine is the major phospholipid present in the biological membranes, and this is about the phosphatidylcholine. And now coming to the, of course, this phosphatidylcholine also, it has used as a therapeutic agent or the supplement to treat various disorders. And then coming to the phosphatidylethanolamine, and in this the phosphatidic acid is further estrified with the ethanolamine. You can see here, and this phosphatidylethanolamine is also known as the cephalins, and the cephalin is mainly present in almost all living cells. Mainly, particularly it is present in the nervous tissue, that is the white matter of the brain, and also the nervous tissue. And these cephalins are the principal in the bacteria as well. This is the principal phospholipids in the bacteria also. And then Then coming to the phosphatidylserine. Phosphatidylserine, as the phosphatidic acid is further stripped with the serine molecule, and here this is known as the phosphatidylserine, and this phosphatidylserine and phosphatidylethanolamine and the phosphatidylserine both are also known as the aminophospholipids because they are aminophospholipids, and this phosphatidylserine is mainly present in the inner leaflet of the biological membrane. So because we know that this is the lipid bilayer, and this is the lipid bilayer, it has the inner half of the bilayer. This is the inner leaflet of the bilayer, and this is the outer leaflet of the bilayer. So the phosphatidylserine is mainly present in the inner leaflet of the bilayer, and uh, and you can also see here that the phosphatidylcholine is mainly present on the outer leaflet of the bilayer, and phosphatidylethanolamine is mainly present in the inner leaflet of the bilayer, and the phosphatidylserine is also inner leaflet, and the of course the sphingomyelin is a outer leaflet. In the sense, all these phospholipids are distributed regularly in the plasma, in the lipid bilayer. So some are present in the inner leaflet, or some are present on the outer leaflet of the membrane. And then phosphatidylinositol. So this phosphatidylinositol, as I already said, that when the phosphatidic acid is further estripated with the inositol, that is a polyalcohol, and the phosphatidylinositol is formed. And this phosphatidylinositol, as I already said, that this phosphatidylinositol is a source for the phosphatidylinositol diphosphate and the phosphatidylinositol triphosphate. They are involved in the signal transduction. They are the messengers. So this is about the phosphatidylinositol. Then coming to the, so these are all the about the phospholipids. And coming to the, in addition to these phospholipids, are the, we have some other noteworthy phospholipids, and these noteworthy phospholipids are the dipalmitoyllecithin and cardiolipin and lysophospholipase, as well as the plasmalogens. So these we will discuss in the next class.